are 3 times more likely to contract cancer than non-diabetics. These facts, which have puzzled medical investigators for years, at last can be explained in light of the trophoblastic thesis of cancer. But what happens if the pancreas is weak, or if the rate of cancer growth is so high that the enzyme trypsin can't keep up with it? Then what? The answer is that nature has provided a backup mechanism, a second line of defense that can do the job even if the first line should fail. It involves a unique chemical compound that poisons the malignant cell while nourishing all the rest. And this is where the vitamin concept of cancer finally comes back into the picture. The chemical compound in question, of course, is vitamin B17, which is found in those natural foods containing nitrillicides. It's known also as amygdalin, and as such has been used and studied extensively for well over 100 years. But in its concentrated and purified form, developed by Dr. Krebs specifically for cancer therapy, it is known as Laetrile. For the sake of clarity in this presentation, however, we shall favor the more simple name, vitamin B17. The B17 molecule contains 2 units of sugar, 1 of benzaldehyde, and 1 of cyanide, all tightly locked together within it. Now, as everyone knows, cyanide can be highly toxic and even fatal if taken in sufficient quantity. However, locked as it is in this natural state, it's completely inert chemically and has absolutely no effect on living tissue. There is only one substance that can unlock this molecule and release the cyanide. That substance is an enzyme called beta-glucosidase, which we shall call the unlocking enzyme. When B17 comes in contact with this enzyme, not only is the cyanide released, but also the benzaldehyde, which is highly toxic by itself. In fact, these two working together are at least 100 times more poisonous than either of them separately. The unlocking enzyme is not found to any dangerous degree anywhere in the body, except at the cancer cell, where it always is present in great quantity. The result is that vitamin B17 is unlocked at the cancer cell. It becomes poisonous to the cancer cell, and only to the cancer cell. There's another important enzyme called rhodinase, which we shall identify as the protecting enzyme. The reason is that it has the ability to neutralize cyanide by converting it instantly into byproducts that actually are beneficial and essential for health. This enzyme is found in great quantities in every part of the body except the cancer cell, which consequently is not protected. Here, then, is a biochemical process that destroys cancer cells, while at the same time nourishes and sustains non-cancer cells. It's an intricate and perfect mechanism of nature that simply couldn't have been accidental. There's much speculation today about carcinogens, the things that supposedly cause cancer. We are told that researchers now have proven that smoking, or excessive exposure to the sun, or chemical additives to our food, or even certain viruses, all can cause cancer. But as we have seen, the real cause is an enzyme and vitamin deficiency. These other things merely are the specific triggers that start the process. Anything that produces prolonged stress or damage to the body can trigger off the production of estrogen as a part of the healing process. If this goes unchecked because the body lacks the necessary chemical ingredients to fight back, then the result is cancer. Specific carcinogens, therefore, do not cause cancer. They merely determine where it's going to occur. Of course, nature's defenses against cancer include more than just the pancreatic enzymes and vitamin B17. Research has shown that an important role also may be played by other enzymes, other vitamins, oxygenation of the blood, pH levels, and even body temperature. Vitamin B17 seems to be the most vital and direct acting of all these factors, but none of them can be ignored, for they're an interlocking part of the total natural mechanism. Fortunately, it's not necessary for man to understand fully every theoretical aspect of this mechanism in order to make it work for him in practice. All that he really needs to know is the necessity of eating foods rich in all the vitamins and minerals, and of minimizing damage or stress to the body. The reality of the vitamin B17 concept of cancer has been proven in the laboratory beyond any doubt. For example, Dr. Dean Burke, head of the cytochemistry section of the National Cancer Institute, has reported that in a series of tests on animal tissue, the B17 had no effect on normal cells, but released so much cyanide and benzaldehyde when it came in contact with cancer cells that not one of them could survive. He said, "When we add Laetrile to a cancer culture under the microscope, we can see the cancer cells dying off like flies." We've said that vitamin B17 is harmless to non-cancer cells. This is true, but perhaps it would be more accurate to say that it's as harmless as any substance can be. After all, even life-essential water or oxygen can be fatal if taken in unnaturally large doses. And this is true also of vitamin B17. For example, there's one case of a man who reportedly died from devouring almost a cup of apple seeds. Incidentally, the case never has been authenticated, but assuming it's true, if the man had eaten the apples also, he would have obtained enough extra rhodonese from the whole fruit to offset the effect of even that many seeds in his stomach. But that would have required that he eat several cases of apples, which of course would have been impossible in the first place. Nature can do only so much. It cannot anticipate excess of this kind. Therefore, it's wise to follow the simple rule that one should not eat at one time more seeds than he likely would consume if he also were eating a reasonable quantity of the whole fruit. This is a common sense rule with a large safety margin that can be followed with complete confidence. Now, when it comes to the laboratory forms of vitamin B17, known as amygdalin or Laetrile, there's even less cause for concern. For over 100 years, standard pharmacology reference books have described this substance as non-toxic. After a century of use in all parts of the world, there never has been a single reported case of related death or even serious illness. In one series of tests, white rats were fed 70 times the normal human dose of Laetrile, and the only side effects produced were greater appetite, weight gain, and superior health, just what one would expect from taking a vitamin. Aspirin tablets are 20 times more toxic than the equivalent amount of Laetrile. And in fact, Dr. Burke of the National Cancer Institute has demonstrated that Laetrile is even less toxic than sugar. Let's turn now to the all-important question. Does Laetrile, or vitamin B17, actually control cancer in human beings? And if it does, is there statistical evidence to support that claim? Spokesmen for organized medicine say no. Almost all official opposition to Laetrile is based upon a 1953 report by the Cancer Commission of the California Medical Association. The report said flatly, "No satisfactory evidence has been produced to indicate any significant cytotoxic effect of Laetrile on the cancer cell." Using this report as a primary reference, government agencies soon declared that it was illegal to prescribe, transport, or even recommend Laetrile. The report was written by the committee chairman, Dr. Ian MacDonald, and the committee secretary, Dr. Henry Garland. There were 7 other prominent physicians appointed to the committee, but none of them, not even the men who wrote the report, had any personal experience with Laetrile. They had based their conclusions entirely on the written records of other experimenters. The scientific judgment of these men perhaps can be best appreciated by noting that MacDonald and Garland were the doctors who had made headlines claiming that there was no connection between cigarette smoking and lung cancer. In fact, MacDonald had claimed that 24 cigarettes per day was a harmless pastime, and then he said, "A pack a day keeps lung cancer away." But even more important than this scientific ineptitude is the fact that both men actually had falsified their summary of the Laetrile experiments. For example, their report claimed that microscopic examinations of tumors taken from patients treated with Laetrile showed no evidence of beneficial chemical effect. Yet, 10 years later, it was learned that one of the pathologists conducting the examinations, in fact, did report several instances of tumor destruction, which he stated at the time could well have been caused by the action of Laetrile. MacDonald and Garland had not told the truth. The report also stated that laboratory technicians had tried unsuccessfully to release cyanide from Laetrile. This was offered as powerful evidence that the entire theory was a fraud. And yet, just 2 months prior to that, the American Medical Association Chemical Lab had reported that it had been successful in releasing cyanide from Laetrile. And of course, other labs have done this also, including the California Food and Drug Lab, and of course the cytochemistry lab of the National Cancer Institute. Again, MacDonald and Garland had obscured the truth. Another important aspect of this report is that the patients had received extremely small doses of Laetrile, much too small to prove anything. Today it's not uncommon to administer 2 or 3 grams of the material in a single injection. Generally 30 or 40 grams are